Kia ora koutou, ko Tiffany taku ingoa, he kairuruku tau whanga ahau ki Manaki Whenua. Good morning everyone, my name is Tiffany and I am the Events Coordinator at Manaki Whenua Land Care Research. Before I hand over to Hugh, I'm going to run through a couple of technical slides to ensure that your experience with us online today goes as smooth as possible. If you have joined us previously for a webinar session, you can ignore me for the next minute. You will notice you have a control panel at the side of your screen. If at any time this collapses, you can bring it back by simply clicking the orange arrow button. If you are having sound issues and you can see my mouth moving but cannot hear a word I'm saying, please grab the PDF in the handout section, and this has instructions to resolve this quickly. The audio panel is where you can control where the sound plays on your computer. Select the drop-down arrow and choose your audio output. During the presentation, you may have questions that you want to be covered in our Q&A session. You can do this via the chat panel throughout our session today. You will notice it is pretty small and it will be hard to read other attendees' questions. Select the pop-out icon on this panel and drag the corner out to make it as big as you want. You can also use this feature if you're having technical issues and ask me any questions. Questions asked by the audience show as anonymous and a green colour in the chat panel. However, please note, we will use your name in the Q&A session. If I respond to you regarding a question, this will show as red. On a personal note, I would like to thank Hugh for his contribution to our webinar series. If you have joined us throughout the week, you would have seen the great job he has done at facilitating these sessions. There has also been a large group of Manaki Whenua staff who have contributed to making this week a success. It has been no easy feat turning a one-day in-person conference into a week's worth of webinars, so please give everyone involved a virtual applause. Now over to Hugh to introduce you to our 10th session for the Biosecurity Bonanza series.
Kia ora koutou everybody, and thank you very much for that, Tiffany. It's been a pretty amazing week, I have to say, hosting these webinars and introducing the people who've been the presenters from our biocontroller weeds team. I've been in the biocontroller weeds team at Manaki Whenua for nearly 40 years now, and I've run a number of these biosecurity bonanzas in terms of live things, and this webinar one seems to have been very successful process and great participation. So it's really great to have you all on board and being a part of this. And today, for the last one that I am hosting, and our last presenter in terms of the biological control is Bob Brown. He's one of our, well, he is our one and only hero working on wasps and wasp biocontrol. Bob is famous for his viral video collecting a wasp nest while being attacked by hundreds, probably thousands actually, of wasps at the time. His video went viral across Facebook and elsewhere, and he became an instant hero, looking for a knighthood, I think, at the time. But today, Bob is here to tell us what he has been up to lately with his wasp biocontrol work. Take it away, Bob.
Cool. Thanks, Hugh. I don't know about the hero malarkey, but yeah, I've been working on wasps now for a little over 10 years, and it's been really fun. So I'm going to talk a little bit about what's been happening in the biocontrol space of the wasps, and I want to start with this first slide is basically just to acknowledge all the people that contribute to this project. And you can see that there's quite a range of contributors, from councils to primary industries and MPI, of course, since this is one of the last Sustainable Farming Fund projects, I think, that was funded. Now that it's changed a little bit in scope to the Sustainable Food and Fiber Futures. So yeah, you can see that, like I said, that it goes right across the gamut from forestry to councils, and so there's also other impacts besides primary industries. So there's also a lot of issues with the environment, but also the human health issue. Some data from ACC shows that there's roughly about 1,000 claims from wasp stings each year. So that's a lot. So why are Vespula wasps so invasive in New Zealand? And one of the main reasons is that they've got a long overwintering diapause, but unlike a lot of other insects, they can break the diapause, come out, have a feed, and then go back into diapause. And this is great for making long trips overseas and colonizing new areas. Also, their biology. So they're social, which means that they've got a colony and different castes. So you've got some wasps that specialize in foraging for food, others for protection of the nest, and others for reproduction. And with Vespula, they're generalist predators, but they're also scavengers. So unlike some of you guys might be familiar with Polistes, that the little paper wasps that are becoming more of a nuisance lately, but those aren't scavengers. They only go for live food, whereas the Vespula will go scavenge roadkill or dog food or cat food or anything like that. So they're very flexible and they can learn. The New Zealand environment is also key to this. So we've got mild summers, mild winters, and it's basically paradise for these guys. And another large contributing factor is the honeydew. So not only is there the honeydew beech forests in the top of the South Island, but recently there's been an incursion of these guys here. These are the giant willow aphids, and this is on, I'm not sure which willow this is, but this was just taken a couple days ago in Lincoln, and there's a whole stand of these small trees that are covered with these willow aphids, and you can see this worker here that has been scavenging for honeydew. So like I said, they're quite adaptable to different food sources. Also, there was an open niche whenever they arrived here, starting in the 1940s. So there's no native social wasps, there's no native social bees in New Zealand. So there was nothing really to resist them sweeping through the country. And since there's no native or endemic analogs, there was no native natural enemies to prey on these guys. So that's where I'm interested in this. So a few years ago, we discovered that there was a mite that was in nests that were kind of, they seem to be about a third or a quarter of the size of a normal healthy nest. And so we started a project investigating, okay, well, maybe these are kind of an equivalent of varroa mite that will attack wasps like varroa mite does honeybees. Turns out not to be the case. They seem to be relatively harmless in the wasp colonies and perhaps even commensals. So they might be providing a benefit by cleaning up fungus or any kind of decaying matter in the nest. However, they also seem to be potential vectors, particularly for aspergillus, which is kind of like well, you commonly see it as a bread mold, but that fungus is pathogenic to wasps and several other insects. But it's also one of the kind of biota of mite guts. So what you can see here in this photo, this is a wasp larva, and she was starting to spin the silk on her pupa, and you can see a lot of these mites here. And what happens in a wasp nest is that these larvae will be prompted to regurgitate some food for worker wasps. And it looks like here in this photo that these mites are feeding from that excretion. And so that's a possible pathway for the aspergillus or other viruses to get into the wasp colony. So from there, the other workers might feed from this wasp and share it around the nest, just because of how they share food in the colonies. We kind of backed off of this because we started to see a lot of diseases that are also common in honeybees. And so since we didn't want to promote anything that may be detrimental to honeybees in any way. We just kind of have put that on the back burner. Another species that has been adventive is Mellet. So these are little tiny wasps. They're only about 1 millimeter or so long. And these are uncommon parasitoids of Vespid wasps. But whenever they do find them, they seem to they seem to like it in this case, this poor wasp here. So you can see that there's several different generations of these Mellet on this Vespula vulgaris here. And you can tell that by the size of these different larvae. I'm assuming that you guys can see my pointer on the screen. And then there's a larger one here. Their primary hosts are leafcutter bees, megachile, or mason wasps. And there is some evidence that they may also go for bombus. So again, this isn't something that we want to promote. In fact, we don't want to promote it at all because of the benefit of megachile and mason wasps as pollinators in the primary industries. Suika faga is another natural enemy, but this one was purposefully introduced, unlike the previous two. There was 3 species that were we attempted to introduce from the 1980s through the 90s, and these are specific to social wasps. One benefit is that they can have many generations per year and a turnover of about 11 days to 14 days. And this is a great photo from my friend Rupert from a recent trip to the UK. And this is a what's called a brachypterous form. So this female has very short wings. So her purpose is to stay within the nest and to basically just infect or parasitize as many of the wasp pupae as possible. There's another form that has long wings, and those are the ones that spread from nest to nest. So for some reason, these have been considered to be unsuccessful introductions in New Zealand, and we're not really quite sure why that is. We suspect that low genetic diversity is one of the key components, because there's some talk that basically all of the ones that have been released derive from 1 or 2 females that were reared in containment. So that's probably a contributing factor. There's also the possibility of range disparity. So the ones that have been released originated from Switzerland, and we now know, we'll talk about that in a minute, but we now know that the wasps in New Zealand actually come from the southern part of England. So it could be that they're adapted to the wrong ecotype. I was thinking that the cuticular hydrocarbons, the wax that covers the surface of the adult insect, could be a way that the wasps are detecting these parasitoids. But we've just done a recent paper that showed that the Suika faga are able to change their cuticular hydrocarbons on the surface of the body to match at least a couple of the compounds, depending on whether it's in Vespula germanica or Vespula vulgaris. So that pretty much poured water on that idea. So I noticed in some of the other talks there were quite a few questions about the process of identifying new agents, and so I just wanted to talk a little bit about that. So one of the first things that we need to figure out is where this invasive species comes from. And Phil Lester's group up in Victoria Uni have done the heavy lifting on this. So they were able to find that both species of Vespula, the common and the German wasp, both originate from southern UK. And from there, now that we know that the origin, we have to do probably the least glamorous part of the job is literature review. So there's a lot of a lot of literature out there to look through and to get some, kind of a detective story and try to figure out what might be out there, what might be available, and get an idea of how these will interact, essentially. From there, we figure out the likely host range. So you can do this by what's called centrifugal phylogeny, where you have the taxonomy of the target, and then you have, and from there you kind of spiral out from there to see what the nearest relative is in the invaded range. And also similar ecology, what social, in this case, what's a social insect, ground or cavity nesting. And one of the great things about these wasps coming from the southern UK is that they've got a long history of naturalists that have, for some reason or another, have just been documenting host ranges or not host ranges, but ranges of these things and when they're occurring, and basically just trapping for different species. So one good resource for me was the Bees, Wasps, and Ants Research Society, and then there's another one, the Hoverfly Recording Scheme. And so they're really good sources of knowledge on when things occur and where have they been found in the UK. And so one of the last things we do is to go there and survey, get your feet on the ground and actually look in the nest and see what occurs in there, which for me is the fun part. So on this slide, we've got our targets picked out. So we've got Vespula germanica and Vespula vulgaris. So what else is related that's here in New Zealand? So there's one other solitary wasp, the Ancistoceras gazella, which is the European tube wasp that you might see in your garden if you put out those little bamboo or straw bee houses. They'll come through and cover that with mud and lay their eggs in there. Another 3 paper wasps, the Polycis chinensis, dominula, and humilis. The humilis is from Australia, chinensis is from Asia, and Polycis dominula is the European paper wasp, which is a recent addition to New Zealand. And I think this is the one that is really kind of causing some headaches up in the Blenheim area, Blenheim and Nelson area. So they have a weird preference for nesting right under your roof awnings, and also in used cars or something like that. So you go to a car lot and you go to check out the car and you open the door and then all these wasps fly out. So it's pretty interesting. But there's no native species in this family in New Zealand, so that makes the host range testing a lot easier and a lot more straightforward. Whereas if we wanted to look for biocontrol agents, say to introduce to the US or Canada, where they have a lot of native species, then it would be a lot more difficult. It would be a lot more intensive of host testing. As far as similar nesting ecology, bumblebees are probably the nearest relatives, and of those, there's a Bombus terrestrius is probably the easiest to get a hold of because you can get them available commercially. So after doing the host or the literature search, Volucella and Annis quickly came to the top of the pile for me. One was that it's got a great host range, or an excellent range match, so that covers a lot of the southern part of the UK. It was debated for a little while, but on if it's actual parasite or commensal. of Vespula nests, but there's clear evidence that it actually is a parasite of Vespula, and I'll show you some video clips here in a minute. These were also found in nearly every nest that was surveyed in the trips to the UK. They've got a high fecundity, so they average around 300 and some odd eggs per female, but some wild caught ones had over 600 eggs. And being hoverflies, they're excellent flyers, and this would be beneficial for a rapid spread if they are able to be released. So here's the life cycle of these guys. So we'll start with the adult hoverfly here, and so these will fly into the nest entrance of the wasps, and she will either lay her eggs on the envelope of, I'll show you a diagram here in a second, but the nest actually sits inside of an envelope that goes around it. And so she will fly in and lay her eggs on the outside of that envelope or in the nest tunnel. These will hatch and into first larval instars and go in to the comb and start to feed on wasp grubs. Now this isn't exactly like a parasitoid, so they're more parasites at this stage. So they will wiggle in and pierce the skin of the wasp grub and feed from the hemolymph, essentially the insect blood, and like little vampires. And then they'll keep doing that, and they'll move to different wasp larvae until the end of the second stage. And from there, they will look for a wasp larvae or grub that is just about to pupate. So that's when they spin the silk over their cell, and then they change from a larva to an adult in there. And so what the Vespula does is that it waits for that the larva to start spinning the silk, and then it's shut the door essentially, so nothing can come in, nothing can get out. And from there, it transforms to the third larval stage and will consume all of that wasp grub. And from there, it will exit the cell, usually by pushing a hole through the side of the paper, and then it will go to find an overwintering spot in the soil surrounding the nest. So here's a sketch I did of a wasp nest. So this is the entrance tunnel. This is the main comb here. Let's see if I can. So this is the entrance tunnel, and here's the envelope that surrounds the outside of the nest. So this is essentially how they control the conditions of the nest. So it's like a heating and air conditioning setup essentially. So it's insulation, but also ventilation. So the Vespula will fly in, lay her eggs, usually around here or sometimes here, and they will hatch and find their way into the inner chamber, and we'll go and find some grubs. So this is, in this photo we've got, so these are wasp grubs, and we have 1, 2, 3 Vespula inanus, second larval stage. And you can see that they're dorsal ventrally flattened so that they can fit in the cell between the cell wall here and the larva. And what they do is they wriggle their way down and feed from below. So here's a video. Hopefully it plays all right. There we go. So this is a Vespula second instar larva. It knows it's been rumbled because this was whenever I was digging up a nest, and it's running for the nearest hiding spot. And you can see over here that there's another one, and here's one that is actually under the silk cap. So it's that one's getting ready to transform into the third larval instage, and from there it will devour that. And here's another nest that has quite a few Vespula in it. It's pretty heavily infested. This is the stuff of wasp nightmares. But it's what I like to see whenever I dig a nest up. So another one that came up during the literature review was the Metaleucus paradoxus. Now these have quite a bit different biology than the Vespula, but they are also a brood parasite. So they attack the immatures of the Vespula. These tend to have a preference for Vespula vulgaris, which I'll show you in a little bit. And they were found in quite a few nests in the UK surveys. They've got a fecundity, a really high fecundity of up to 700 eggs per female, but their life history is pretty complex. And this was one of the reasons why kind of originally it was overlooked as a potential biocontrol agent, because they thought it was a little haphazard. So what happens is, so this is a female, or this is a male here that you can see that because it's got that really ornate antennae. So these guys leave the nest. They don't live for very long, the adults. They mate, and then the female will lay eggs on wood that is suitable for wasps to gather for pulp. And so then the wasps will go there next spring, summer, and autumn, and gather these eggs, or gather the pulp. And from there, the eggs of these guys will have hatched into little tiny larvae that will then jump onto the workers, and the workers will carry them back to the nest. And as they're building the nest, the triungulin larvae, which is what these are called, they will jump off of the worker and get into the nest. And I'm not sure if they actually are fed to the wasp grub, or if they puncture a hole and enter the wasp grub through the dermis. But they are internal parasites for the first stage. And yeah, here we go, the life cycle. So the first stage is inside the larvae, and then they punch their way out, and then they shed their skin, and that clogs the hole where they punched out of the wasp larvae. And from there, they feed on the exterior of the wasp. And during this, they consume the whole wasp again. And here's a photo of one that we found. So this is what a normal Vespula larva or pupa would look like, and this one has been parasitized by a Metaleucus, and so this one's in the process of basically just sucking all the life essence from this pupa. So over the past few years, we've been going to the UK to survey nests, and this is what we found. Basically, very close. They've all been highly parasitized, mostly by Vespula, but vulgaris, the common wasp, has also been about half of the nests have been parasitized by Metaleucus, and only a couple have been parasitized by Suika faga. So this is a pretty low number. This is kind of, you could do 30 nests sometimes in a day here in New Zealand. So this also shows you how the low population of the nests of these wasps in the UK. So I had read in the literature that the Metaleucus had also parasitized Germanica. So I kind of combined a couple of previous papers with what we found in our surveys, and so that gave us a total of about 420 nests surveyed, and still a rather low percentage of Meadowicus going for Germanica nests. And so,
Bob, I just want to briefly interrupt you there, sorry, but
yep.
you've got a couple of minutes left to go. Is that all right?
Yeah, that's fine. Yep, sorry.
Cool, thank you.
Okay, thanks. So what I found was interesting was that on these surveys, we found that Suika faga was really rare. And so kind of what you want as a biocontrol agent is that you want something that is relatively common, but whenever you introduce it to its new range, that it'll be released from its own natural enemies, and then therefore have a large impact on the target species. So these are very common across both species, and this is pretty fairly rare. But that could also be that since I was over there collecting nests towards the end of the year, that that might be an artifact of the surveying. So part of the research, I got in touch with the National Bee Unit over in the UK, which is part of the Animal and Plant Health Agency, and they do the surveys of honeybee hives, and they have done so since the 1950s. So I was trying to get access to their data, but they said that no, I couldn't have it. But what am I looking for? And then they'll check for me. So I got in touch with them, and they ever so kindly told me that no, the species that I'm looking for haven't been found in any of the nests, and they've been surveying about 35,000 nests or hives per year since 2010. And they also told me that they do have specimens of Volucella and Meadowicus as reference samples to look for in those nests. So that's very good. So about 350,000 nests have been surveyed over the last 10 years. This year will be a lot lower, I'm sure, because of the pandemic. And I also host tested some Bombus terrestris audax while I was over there, and there was no attack on brood. So just quickly, next steps. We're in the late stages of writing the application to submit to the EPA. The next one is a bit of a bone of contention with me. I'm developing rearing methods. I've had a lot of problems with rearing these species recently, so I need to do a lot better on that. And yeah, hopefully get to release them. And also there's some other potential biocontrol agents, but there's little to no information on those. So I'm keen to look at some other species. And on the right here, we've got the kind of timeline, the flow chart, if you will, of the process of applying to the EPA. And these are the days that you can expect each process to take. So, okay, that's it for me. Thank you.
Cool, excellent, Bob. Thank you very much for that. You can tell just how passionate you are about your topic and how much you like to talk about the work that you're doing. And it really is fascinating stuff, I have to say. And the sooner we can get something working on wasp biocontrol, the better, because there's certainly a pain in the butt of things to have around our environment. There are a few questions, so I'll be reasonably quick with these. Okay. But the first one's from Graham Surrey. And his question is, how does the adult Volucella make it inside the wasp nest without being attacked by the sentry wasps?
Oh, well, that's a good question. So there are some that get attacked by wasps. So every once in a while you'll find a dead one in the bottom of the nest. So that didn't quite make it in. But they are, I don't know if I can pull up the picture here, but they are pretty good visual mimics. So, oh, sorry, there we go. So they do look a bit waspy. So yeah, the funny thing is that there's another species of Volucella that isn't a parasitoid, but it is a commensal in the bottom of the nest that looks nothing like a wasp, and they can fly in without any problem whatsoever. They're just black and white, and so I don't know if it's chemical or sound, but they don't seem to be bothered at all by that.
Okay.
So,
another question is from Jesse Fairbrother Lambert. Regarding Volucella, could they compete with native species when it comes to food the adults eat?
possible. It's going to depend on phenology and that sort of thing. These are, yeah, they're generalist kind of nectar feeders. So they will definitely compete in that aspect, but to what extent, I'm not sure. Yeah, there's already a lot of pressure in that area from honeybees and bumblebees, and
Sure.
Yeah. But if there's not very many wasps, there won't be very many Volucella. So
Yep, true enough.
If you see a lot of Volucella, that means that they've killed a lot of wasps. So
Yeah, exactly.
yeah,
And soon to be a lot less Volucella. So. Right, Sarah Jackman has the next question. How can you tell when the Volucella larvae are under the silk cap with the wasp larva?
well, I don't know if it was, I don't know how well the video came through, but you could actually see on the underside of the larva, they've got these little claws, and you can see those gripping onto the silk cap, and you can see it moving under there.
Yeah,
but,
I imagine you know what to look for, but the rest of us probably weren't quite so sure what you were showing us.
Yeah, sorry. Yeah.
It wasn't that often. No, that's all right. It's not a apology. It's just that it's hard to see because, yeah, not as obvious for us, but cool.
Yeah,
Good
but
to know.
also whenever I go through and whenever I collect a nest, I will open up some of the capped cells just to have a look to see what's in there as well. And that's when you find them lurking under there.
Okay. Graham Surrey's asked another question. What are your thoughts on the risk of the Asian giant hornet, the wonderful Asian giant hornet, incursions into New Zealand, given their recent discovery in the US? Do we have any idea of the possible incursion pathways for this species?
Yeah, so they have been found previously in used vehicles from Japan, as far as I understand, but they were detected on that case. As far as the giant hornet, I'm not as, I mean, obviously any new incursion will be bad, but I think probably the most likely will be the yellow-legged hornet that is actually spreading through Europe and also through Asia. So it's kind of spreading on two different fronts, and that one seems to be really difficult to deal with in France, and now it's kind of got a foothold in the UK as well. But yeah, that one's really spreading through Europe right now.
Okay. John Hampson says, when do you anticipate lodging the EPA application? I assume next month, is it going to be? Or?
Yeah, soon. Sometime before the end of the financial year is my goal. It was already supposed to be in, but yeah.
Other things get in the way, especially viruses. John Hampson has also asked, what would be the implications if either or both biocontrols were more effective just on the wasp species? Could we just end up with more of one wasp species that are less effective?
I guess what they're asking is, will they compete with each other, the parasites? But I've found in almost all of those nests that were parasitized, I found more than one parasite in there, and so they really don't seem to mind being, you know, in the same thing. And the idea is that the more tools you have, the better, essentially. So the more agents attacking these, the more damage will be done. And so Volucella are more common towards late summer and through autumn. And Suika faga can start from, you know, as soon as they start making the nests in the spring. And the Meadowicus can also start in early summer. They're thought to only have one generation, but it depends on when the wasps will pick them up and bring them back to the nest. So there's kind of a spread throughout the season, and there's also some other species that I'm hoping to have a crack at later on that have distinct summer and autumn phenologies, so that they attack different parts of the cycle, I guess. So.
Of the life cycle.
Yeah.
I'm wondering if John's also sort of wondering about Suika faga and Volucella attack Vespula vulgaris and Germanica, or do they prefer one from another? So
okay, yeah. So
I'm wondering if he's asking that question.
Yeah, so the Volucella and the Suika faga will attack both species equally.
Okay.
The Meadowicus seems to have a preference for Volucella vulgaris, and that is probably due to how they target the wood that the wasps are foraging on. So they lay their eggs on decaying wood in the autumn, and that's the kind of wood that vulgaris likes, whereas Germanica kind of likes more sound wood, I guess.
But I guess what you are saying is that actually all of the species other than Meadowicus, but the other Suika faga and Volucella will attack Germanica
Right.
or vulgaris equally.
And,
so it's unlikely
yeah,
that one of those wasp species.
Yeah, so currently there's, at least in the beech forest, there's about 90% vulgaris and about 10% Germanica, whereas in urban areas it's more equal, so it's about 50-50. So
cool. All right, Mike Cripps has a question. So Bob, you're back again. Any potential negative interaction between potential agents? I think you've just answered that question from John's, but should you just release the single best agent? But actually, you've sort of explained that already, I think.
Yeah, well, I think at the very least, we're going to have an additive effect. Not necessarily a synergistic, but definitely an additive. The more we have, the better. But I don't see any evidence for a subtractive interaction.
Yes, generally the case in terms of biocontrollers, the more things that you can introduce to attack your target species, then the better the control, the level of control and the impact you can have. That's certainly true. And they generally tend to be accumulative rather than competitive too. So.
Yeah, and they might have a preference once they're released on habitats that they like over, so yeah.
True. Okay, looks like the last question that we have at the moment from Nick Goldwater. Are there any viruses that show potential for controlling wasps?
Yeah, but there's also potential for controlling honeybees. So the problem is that there's a lot of overlap with pathogens between honeybees and the wasps, and that's mostly due to the fact that wasps will raid honeybee hives, and so they're taking in some of the bee pathogens. But since there is such a close interaction, I'm not comfortable with getting a disease that may be transferred the other way, whereas a wasp could vector something to honeybees. I think that's a bit too risky, unless we have something that is definite. and that's not my area of expertise. I mean, other people might be able to come up with something, but that's out of my comfort zone.
It's excuse me, and it certainly would appear from a couple of our earlier talks this week from our molecular biocontrol people that there's not enough known about viruses for us to be able to predict which are symbionts and which are not, and which ones will be useful as biocontrol agents and which ones aren't, and how many strains there are and all those kind of things. So I think there's a lot more work needs to be done on that kind of stuff first, I would think.
Yeah.
Excellent, Bob. Well, thank you very much for a really interesting talk. Really great.
Sorry, I went over.
Yeah, no, it's great.
I get on a roll. There's nobody to tell me otherwise, so yeah.
It is a bit like that. I do want to thank everybody for attending these webinars that we've had this week. I also want to thank Tiffany and Manaaki Whenua for allowing us to put these on, and for Tiffany educating us all on how to do this and being the technical person behind it all. I should also say that the mushrooms that are all behind me, which several people have commented on, are actually my wife's, and they're collected from around the world, and there's kind of a story associated with each one of them, but it gives you something else to look at if you get really bored with anything else that you can see. So it's kind of what they're all about in the background. But yeah, again, thank you very much everybody attending. We've had a really good week with lots of really interesting talks and lots of really good support. So thank you, Tiffany. Thank you, Bob. Thank you, everybody else.
Thanks, you.
That's it. Cheers.
Cheers. See ya.